Hi everyone, so for part 2.2 of the module, we will be taking a look into how early life gut microbes may change over time. We will also be looking at how changes due to different exposures early in life may impact on health. So in part 2.1 of the module, we've touched on mode of delivery as an early life exposure, which may modulate the infant's gut microbiome. Since we might already be acquiring our microbes in utero, a number of prenatal exposures have also been shown to modulate these microbial profiles. For example, gestational duration, prenatal antibiotic treatment, maternal prenatal stress, as well as maternal diabetes status. Other early life exposures, such as breast versus formula feeding, the use of antibiotics, and the age at which solids are introduced, may further modulate our gut microbial profiles. But why is this even important? Well, caesarean section delivery compared to vaginal delivery, for example, has been associated with an increased risk for immune disorders such as allergic rhinitis, asthma, and celiac disease. And due to the health benefits associated with vaginal delivery, studies have now started to explore the possibilities of, for example, vaginal seeding, also known as microbirthing. This is a procedure whereby vaginal fluids, and hence vaginal microbes, are applied to a newborn child delivered by caesarean section. It involves placing swabs in the mother's vagina and then wiping them onto the baby's face, mouth, eyes, and skin. Dominguez Bello and colleagues, for example, have shown partial restoration of the gut microbiota of caesarean-born infants via vaginal microbial transfer. However, vaginas don't only harbor beneficial bacteria. There are villains among the heroes, and swabs don't discriminate. The most common causes of neonatal sepsis are organisms that the infant picked up on the way out. For example, Group B strep. Mothers in the US are routinely tested for this bacteria, and about 30% of them have it. Group B strep lives in the vagina without causing any problems, but it doesn't play nicely with babies. It's a major cause of neonatal bloodstream infections and meningitis. Mothers who have been found to carry this bacteria are treated with antibiotics during labor, an intervention that has reduced the rate of neonatal sepsis and saved a lot of babies' lives. Then there's breastfeeding versus formula feeding. Breastfeeding has been described as the most relevant postnatal factor that supports an adequate microbial colonization and drives immune system maturation. Compared to formula feeding, breastfeeding has been associated with decreased morbidity and mortality in infants. Breastfeeding has also been shown to lower the incidence of gastrointestinal infections and inflammatory, respiratory, and allergic diseases. Findings from 16S rRNA high-throughput sequencing studies have now even resulted in clinical trials determining the effects of a probiotic-supplemented infant formula on the healthy human intestinal microbiome during the first year of life. Antibiotics, on the other hand, also modulate our gut microbial profiles and may result in a dysbiotic or disease-associated microbiome. For example, a study by Rob Knight's lab showed that an infant fecal microbiome was more similar to the maternal vaginal or skin microbiome around the time of delivery compared to the maternal oral or fecal microbiome. They then followed this infant over time and showed that his or her fecal microbiome became more like the maternal fecal microbiome as the child aged. They then showed that upon antibiotic administration, the child's fecal microbiome regressed towards the more infant-like profile. Later, they observed a recovery in the infant's fecal microbiome, again progressing towards the adult fecal microbiome. Finally, at around 2 years of life, the child's fecal microbiome was more adult-like than infant-like. By following this child's fecal microbial progression over time, with exposure to an antibiotic, shows the dramatic impact of antibiotics on the fecal microbiome. Every time an antibiotic is administered, it has the potential to modulate our gut microbiomes, which may predispose us to disease. Antibiotic-induced alterations of the gut microbiota may also result in Clostridium difficile infection, which is a significant public health problem associated with increased morbidity, mortality, and healthcare-related costs in the United States and around the globe. Even though antibiotics are the first line of treatment, they are also key risk factors in pathogenesis of Clostridium difficile infection. Fecal microbiota transplantation, or FMT, also known as a stool transplant, has been used successfully to treat recurrent Clostridium difficile infections. This tagline on the screen is the deadly serious tagline of OpenBiome, a US-based stool bank that actively solicits donors at $40 a poop to provide the raw material for its stock in trade, the fecal microbiota transplant. Just because all of this sounds very strange and unfamiliar, I've decided to include a short video from Minute Earth on the topic, which I'm sure you will all find very interesting.
This ecology experiment is one of many that have demonstrated how ecosystems with many species tend to be more resilient than those with few species. We're biased towards this particular grassland experiment because it's run by one of our writers, but strength in species numbers is also key to the health of ecosystems ranging from potato fields to rivers to the inside of your gut. In contrast, when a single species is dominant, it can cause problems. Take the case of Clostridium difficile, a natural part of your gut community that causes a severe inflammation of the colon when it becomes too abundant. This bacteria, often referred to as C. diff, was named difficile because it was difficult to grow in a lab, not because it's difficult to treat, but it is. C. diff infections often develop because these bacteria persist better and regenerate faster than other gut bacteria following an antibiotic treatment for some unrelated health problem. Ironically, the standard treatment for C. diff is yet more antibiotics, which sometimes works, but in many cases the antibiotic resistance of C. diff means the playing field is simply cleared of its competitors and its population can grow even more vigorously. So vigorously, in fact, that of the roughly 500,000 people infected each year in the US, about 1 in 30 die as a result, even after further drug treatments or surgery to remove parts of the colon. Out of desperation came the idea to entirely replenish the gut microbiome, not with probiotics, which only supply something like one living organism for every 10,000 bacterial cells already in your gut and just aren't powerful enough to make a difference, but rather with something more like the microbial equivalent of a blood transfusion. And just like a blood transfusion, don't try this at home. In a fecal microbiota transplantation, or poop transplant, someone with a healthy, diverse community of gut bacteria donates a sample to be administered to a patient with a C. diff infection. And in about 400 of 470 documented cases, the transplanted bacterial community quickly became dominant, causing the patient's digestive system to recover and stabilize, though scientists still don't know exactly how that happens. The human gut isn't the only ecosystem where this germ of an idea is taking hold, either. For example, transplanting soil from healthy fields to unhealthy ones helps knock back crop diseases and make soils more fertile. And transplanting water, snails, and duckweed from a pond to an aquarium helps maintain a well-balanced fish tank. At the moment, microbial transplant therapy for disease control resides at the fringes of medicine, agriculture, and home fish tank husbandry. For medicine, this may in part be due to people being squeamish about their own and others' fecal matter, and insurance companies being squeamish about covering the cost of the procedure. But money also plays a broader role. New practices, even really good ones, have difficulty spreading without active promotion, and that promotion often comes from places like the pharmaceutical, agrochemical, and home aquarium industries. Industries which promote only those things from which they can make a profit, like drugs, fertilizers, aquarium supplements, and other heavily engineered chemical cocktails. But who will promote the simple solutions? We will. From your C. diff to recoup, choose someone else's poop.
In addition to Clostridium difficile infection, a number of studies have shown that shifts in our gut microbiomes are associated with, for example, irritable bowel disease. For example, Palm and colleagues published a paper in 2014 where they used flow cytometry-based bacterial cell sorting and 16S sequencing to characterize the taxa-specific coating of the intestinal microbiota with immunoglobulin A, IgA-seq for short, and showed that high IgA coating uniquely identifies cholitogenic intestinal bacteria in a mouse model of microbiota-driven colitis. They then used IgA-seq and extensive anaerobic culturing of fecal bacteria from irritable bowel disease patients to create personalized disease-associated gut microbiota culture collections with predefined levels of IgA coating. What Palm and colleagues found was that intestinal bacteria selected on the basis of high coating with IgA conferred dramatic susceptibility to colitis in germ-free mice. So in summary, our gut microbiome is a plastic entity which is modulated by a number of exposures throughout our lives. A large number of 16S studies have contributed to our current knowledge of the gut microbiome, which has led to a number of potential interventions for disease states. To date, the majority of 16S studies have focused on the gut microbiome. This research, however, is still very new and more well-designed studies are needed to better understand not only what's there, but also what we're doing. This then brings me to the end of part 2.2 of Module 2.